Hello everyone, welcome to Get My Class. Let's solve this question which says which of the following statements is true? Option A, in type 2 and type 3 enzymes, one large enzyme complex carries both restriction and methylase activity. Option B says in type 1 and type 2 enzymes, one large enzyme complex carries both restriction and methylase activities. Option C says in type 1 and type 3 enzymes, one large enzyme complex carries both restriction and methylase activities. And option D says in type 1, type 2 and type 3 enzymes, one large enzyme complex carries both restriction and methylase activities. So let us understand the question. It says which of the following statements is true? So we have to look for the correct statement. So let us see the first one. First one says in type 2 and type 3 enzymes, one large enzyme complex carries both restriction and methylase activity. So what are they talking about? Type 1, type 2, type 3? They are talking about restriction enzymes. Okay, they are talking about restriction enzymes. What are restriction enzymes? Restriction enzymes are molecular scissors. Molecular scissors. Okay, and what are these used for? These are used to cut the DNA. They are used to cut the DNA, and of course, they are specific to their restriction site. So the DNA consists of restriction site, okay, which will be recognized by these enzymes, and then the cleavage will be brought about. Okay, cutting will be brought about. Now, based on their function and based on the structure, there are three types of restriction enzymes. Type 1, okay, type 1, type 2, and type 3. Type 3. Now, if you talk about the cleavage activity of type 1, there are two functionals. Actually, there is one core protein, you would say, one main subunit, which will have both the activities. Okay, so one large subunit has both the activities, that is methylation and restriction activity. Restriction in the sense cutting the DNA. Okay, but then it does a random cut. What kind of cut it does? It cuts a random cut. It does a random cut, and usually around 1,000 base pairs away from the recognition site. Okay, from its recognition site or restriction site. Okay, so that is the function of type 1. Now type 2, here the methylation and restriction activities are present on separate entities, separate units, you can say, separate subunits. Okay, separate subunits carry methylation activity independently and restriction activity independently. And these are the most common type of enzymes. Okay, and best example you would have heard about BAMH1, okay, HIND3, etc. These are most common. That's why I'm giving you the example. Okay, now type 3 enzymes, these again are similar to type 1. Okay, but then this will cause it can cause cut in single strand. Okay, it cuts the single strand, and this also has large complex which has both. Okay, the one single large complex which has both methylation and restriction activity. Okay, now of course they all are multimers. That means they do have more than one subunits. But then in type 1 and type 3, the same large complex of protein, or you can say subunit, will carry out both methylation and restriction enzyme on its own. Whereas in type 2 restriction enzymes, they have separate units for methylation activity and separate unit for restriction activity. Okay, so let us see. The option A says in type 2 and type 3 enzymes, one large complex carries both restriction and methylase activity, which is wrong because type 3, yes, but type 1, it has separate entities. Option B says in type 1 and type 2 enzymes, one large enzyme complex carries both restriction and methylase activities. So here type 1 is correct, but not type 2 because as I told you, type 2 has separate entities, hence it is pulled out. Option C says in type 1 and type 3 enzymes, one large enzyme complex carries both restriction and methylase activities. That is absolutely right. And option D says in type 1, type 2 and type 3 enzymes, one large enzyme complex carries both restriction and methylase activity. Okay, But again, here type 2 and type 3, of course, it is correct with respect to that, but with respect to type 2, it is wrong because they have type 2 restriction enzymes have separate entities for methylation and separate subunit for restriction action. Okay, hence the correct option is option C. So let us conclude the answer. The answer is option C. I hope you understood the explanation. Thank you, everyone.